Mallory, and she's going to talk about enhanced alcohol self-administration in rat models of endogenous depression vulnerability versus resistance.
Thank you. Hi, everyone. My name is Mallory Udell. I am a student here in the neuroscience track, and I'm working under Dr. Hao Chin. Today, I will be discussing my project proposal and the data we have so far, which has shown enhanced alcohol consumption in rats with a genetically conferred depression-like phenotype. So I'm sure it doesn't come as a surprise that alcohol is by far the most commonly abused substance. Unfortunately, ever since the pandemic, we have faced an overwhelming rise in the rates of alcohol use disorder, or AUD. In fact, the most recent national survey data showed a US prevalence of 29.5 million cases. This number is more than double what was reported just 2 years prior, back in 2019. We are also seeing a rise in underage drinking, and looking specifically at those between ages 12 and 17, we're finding that adolescent girls are not only engaging in more binge drinking, but they're also more likely to develop an AUD compared to their male peers. And we know that even though majority of the population drinks, there are certain subpopulations that are more vulnerable to substance abuse than others. For instance, AUD rates are particularly high among those with depression. When broken down by age, depression affects adolescents at the highest rates, and having depression more than doubles their risk of binge drinking. Depression is also reportedly more common among girls, who again are also at an increased risk for AUD compared to boys. So it appears that adolescent girls represent a group that is particularly vulnerable to this comorbidity. And I'd also like to note that when these conditions do co-occur, they aggravate one another, making each disorder more severe and harder to treat. We also know that dual diagnoses patients require different courses of treatment, but unfortunately, many are still being prescribed the leading drug therapies for depression alone, which often even has an effect of exacerbating their AUD symptoms. To study this in animals, we utilized 2 substrains derived from the Wistar Kyoto, or WKY, which is a well-established rat model of depression. These substrains were generated through selective breeding based on behavior in the forced swim test, which is an assay that is standardly used to measure depression-like behavior in rats. So this assay, as shown here, involves placing rats in a body of water and then measuring the amount of time that they spend immobile as an indication of behavioral despair. So about 20 years ago, this test was used to identify individual WKYs with the highest percentile immobility scores in this test. Those rats were then selected as first-generation breeders to produce our WKY more immobile, or WMI substrain. And across all these years now of inbreeding, these rats have maintained their depression-like phenotype. These WMIs have actually even been utilized clinically to develop a panel of blood markers that's been able to successfully differentiate humans with depression from those without it. So they are valid genetic models. And the big advantage for us here is that we also have the WLI strain, the less immobile strain, which, despite being so genetically similar to the WMI, these rats do not show depression-like behaviors. And in the forced swim test specifically, they show a much more active coping, struggling kind of response to that kind of stress. So rather, these rats serve as a genetically resistant control for us. So given that alcohol use disorder and depression are both very heritable, we hypothesized that rats with the endogenous depression-like phenotype would also consume more alcohol if given the chance. In addition, because we're seeing trends of increased binge drinking among females, we also expected to see higher intake in the female groups relative to males. So testing these ideas is what I've been working on for the first aim of my project. And to do that, we developed a novel device called Homebrew, which allows rats to self-administer together in social pairs. This is, of course, different from the standard for self-administration studies, which generally involves placing each rat in its own chamber for testing. While this may be conducive for studying other types of drugs, alcohol is a particularly social drug. So we wanted to create a testing environment that better recapitulated the context in which humans tend to drink. How we achieve this is by first implanting a subcutaneous RFID microchip on each rat's head, right above its nose. And so then that way, during testing, each time a rat pokes its nose through the spout compartment there, the rat's unique RFID is scanned by a scanner we have embedded around the opening. And then that way, each subsequent lick is recorded under the proper rat. So here we have the alcohol associated or active spout, which delivers an alcohol reward on a variable 5 ratio schedule of reinforcement. So what that means is that rats are required to lick 5 times on average in order to receive 1 reward. And our homebrew program also enforces a 3-second timeout period in between rewards. And that's just to ensure that rats have enough time to consume each one before the next is delivered. Then over here on the other side, we have our inactive control spout, which is not associated with any consequence. Then briefly for our self-administration protocol, we start exposing the rats to alcohol during adolescence as typical for humans. So 3 to 4 days after we implant their RFIDs, we then give rats access to self-administer a low dose of 5% alcohol, which is available in 2-hour sessions for 5 consecutive days. After that, all sessions are 4 hours long and conducted on intermittent days. So we do Monday, Wednesday, Friday. Then by the 3rd week, we have rats worked up to the final 20% dose, and we just keep running them on that for 5 weeks in order to establish baseline intake across groups. Then just to show an example of the rats' licking patterns, what those look like, here is some data from rats in our WMI strain showing the average number of licks on either spout across sessions. So here we're seeing really similar licking patterns between males and females, where the number of licks on the active alcohol-associated spout increase gradually over time, whereas the responsiveness to the inactive control spout diminishes. So this just reflects the rats learning to discriminate between spouts, as would be seen in any operant paradigm. Then getting into the baseline intake data that we have so far. Here is showing group averages in 20% alcohol intake, which is measured in grams per kilogram of the rat's body weight. So here's including all 21 total baseline sessions that we ran at that dose. And what we're seeing so far is significantly higher intake in both sexes of the depression-prone rats, here in purple. In addition, we are also seeing a trend of higher intake in females versus males of either strain. Then after the 5th week of self-administration at the 20% dose, we then implement a brief 1-week period of forced abstinence from alcohol, after which we then re-expose the rats to alcohol under the same conditions. And this is done as a way to probe for an alcohol deprivation effect, which is considered a model of relapse. So here is plotting group means in intake during the week leading up to forced abstinence compared to intake during the week of re-exposure. I am still working on collecting data for this, but what we're seeing so far is, at least in males of the depression-prone strain, they are significantly increasing their intake from baseline to re-exposure. So this is also notably not seen in either sex of our depression-resistant rats. And even such a relatively short break from alcohol is enough to drive these males to consume larger amounts when they're reintroduced. So this kind of effect is similar to what we would see in humans during a state of relapse. Finally, after the re-exposure week, we test rats through one last week of self-administration. this time on what's called a progressive ratio schedule of reinforcement. So these kind of tests involve increasing the number of licks that are required for delivery of one alcohol reward. This is done somewhat exponentially after each reward is obtained. So what we are seeing so far is a strain difference within males, where males, and I guess I need to define breakpoint here, reflects the maximum number of licks that they're willing to expend for one reward, which is a measure of kind of like the effort they're willing to put in for it. So we are seeing significantly higher average breakpoint across 3 progressive ratio sessions in our depression-prone males relative to the depression-resistant ones. And this indicates that they are more motivated to obtain alcohol than their resistant counterparts. Again, still working on collecting data for this. I actually would have expected to see the highest breakpoint in our depression-prone females, but as we are still increasing our sample size, I'm curious to see how these trends could evolve. And altogether, the data that we have so far does support the idea that alcohol use and depression go hand in hand. And I'm pretty excited about these data because, as I mentioned before, these inbred strains we're studying are nearly isogenic. So because of this, they give us this really unique opportunity to later look into which of their genetic differences are most likely related to the differences we're seeing here in alcohol use behaviors. So I'm definitely excited to take advantage of that opportunity in our next steps once we get this behavior portion wrapped up. And with that, I'd like to thank my lab, especially my wonderful advisor, Dr. Hal Chin. And special thanks to June for all of the work that he has put into developing and troubleshooting the homebrew devices. And of course, special thanks to each of my committee members. And I'll take any questions.